This will be the first of 3 videos on the nervous system, and what we're going to discuss in this video is more the cells of the nervous system than anything else. So the first thing to talk about is the overall organization of the nervous system. And so the nervous system can be divided into 2 major, basically, divisions: the central nervous system and the peripheral nervous system. So the central nervous system is going to be the brain and spinal cord, and the peripheral nervous system is going to be everything else. Now, one thing that I want to point out about this video is if you look to the lower left, you're going to see some keys to really fully understanding this diagram. Within each of the boxes here, you'll see a bullet point with a circle and a bullet point with a square, and the bullet point with the circles are the structures that are involved in these particular portions of the nervous system, and the square bullet points are the functions of those particular sections of the nervous system. When you're looking at the different parts of the diagram itself, then you're going to notice a couple of different things. You're going to notice that there are some lines in blue and some lines in red, and the lines that are in blue are a part of the sensory division of the peripheral nervous system, and you'll notice that signal is actually coming into the central nervous system. The red portions are the motor or efferent division of the peripheral nervous system, and again, you'll notice that signal is going outwards from the central nervous system to the peripheral nervous system. So back to our boxes here, the central nervous system is the brain and spinal cord, and it serves as the integrative and control centers, basically meaning that it takes data that it receives from the peripheral nervous system and makes decisions based on its interpretation of those data. The peripheral nervous system involves cranial nerves and spinal nerves. So basically this is everything but the brain and the spinal cord. And basically what you've got in the peripheral nervous system is communication lines between the central nervous system and the rest of the body. The peripheral nervous system can itself be divided into 2 divisions: the sensory or afferent division and the motor or efferent division. And looking at the sensory division first, it's composed of somatic and visceral sensory nerve fibers. And so somatic nerve fibers are going to be those that are coming from the skin and the skeletal muscles. The visceral sensory fibers are going to be coming from everywhere else. And you can kind of think of this as information that's coming in on body position and what's happening at the surface of the body versus information that's coming in from internal organs. The motor or efferent division consists of motor nerve fibers, and this is sending directions, conducting impulses from the central nervous system to effectors, which are muscles and glands. And so the word effectors basically means something that has some sort of an effect inside or outside the body. So do you want to move? Do you want to digest food? Do you want your heartbeat to speed up? Do you want to breathe? So effectors are structures that carry out some sort of task. And so the motor or efferent division involves sending directions to effectors to tell them whether to carry out their function at all or how intensely to carry out their particular function. The motor division can itself be divided into the somatic nervous system and the autonomic nervous system. And so the biggest difference here between these two is that the somatic nervous system is voluntary, or it's mostly voluntary. So here is where you get impulses being conducted from the central nervous system to skeletal muscles for some sort of voluntary movement. Now, not 100% of skeletal muscle contraction is voluntary. That's actually something that we learned on the last test when we talked about sympathetic tone, but the majority of it is. With the autonomic nervous system, these are going to be visceral motor fibers, and these are going to be involuntary. And so this is going to control a lot of processes that happen, like your heartbeat, your respiration rate, whether or not glands get turned on, smooth muscles that line your digestive tract or your respiratory tract. So these are going to be involuntary impulses to effectors like cardiac muscles, smooth muscles, and glands. Now, the autonomic nervous system itself can be divided into 2 divisions itself. One is the sympathetic division, which mobilizes body systems during activity. Now, we're going to find out a lot more about the sympathetic division in Test 5 material. But this is basically what most people refer to as fight or flight. It mobilizes body systems during activity. The other division of the autonomic nervous system is the parasympathetic division. Now, this is something that does just the opposite of fight or flight. This often gets called rest and digest because it conserves energy, and it also promotes functions like digestion, things that keep your body running in non-emergency situations during rest. And so this is fight or flight, rest and digest. And as I said, we'll learn a whole lot more about this in Test 5 material. So another thing to remember that I want you to remember about the autonomic nervous system is that it does have its own separate architecture. So the nerves that are involved in fight or flight and the nerves that involve rest and digest they're literally separate nerves from nerves that go to the somatic nervous system. Okay, so the major functions of the nervous system are going to be sensory, which senses changes through sensory receptors, integrative, which analyzes this incoming information, stores some aspects, that's what we call memory, and it makes decisions regarding appropriate behaviors. And so once you've made some sort of decision on what to do with the information that you've got, then you send impulses through the motor portions of the nervous system, which is your response to some sort of a stimulus. And I don't want you to think that this just means something like movement. This could be something like raising body temperature in response to an infection, raising respiratory rate during exercise. So these are not necessarily voluntary things. Okay, so the next thing we're going to do is take a look at neurons, and we're going to see different parts of this neuron that I want you to know and understand for the test. So the first portion here is the cell body. This is a receptive region. So one thing that I want you to kind of remember back to is the motor end plate on a skeletal muscle cell. And so the motor end plate was the portion of the skeletal muscle cell that received information from the nervous system. And so in neurons, because you don't ever have a pathway from the central nervous system out to an effector or from a sensor into the central nervous system that consists of only one neuron. You've got multiple neurons that are involved in either getting signals out to effectors or getting signals from receptors into the central nervous system. So because you've got multiple neurons in this chain of communication, it means that you're going to have receptive regions, and those receptive regions, some of them are on the cell body. The other thing that the cell body does is to synthesize the membrane proteins. And so, as we'll see, the membrane proteins are extremely important in terms of nervous system physiology. So, which proteins are we talking about? If you'll recall, again, back to the skeletal muscle physiology, you had voltage-gated sodium channels, voltage-gated potassium channels. You had channels that maintained a resting potential. And so same thing here, because nervous tissue is electrically excitable, you're going to see a lot of the same membrane proteins that we have already seen with the muscle system, with the skeletal muscle system. These are dendrites, and I've got these arrows pointing at the base of the dendrites, but you'll notice that there's some pretty extensive branching. So these are receptive regions, and the branching of these dendrites, what this does is this allows neurons to communicate with many other neurons if necessary. This is the nucleus. It is, of course, the control center of the cell. This is the axon. So the axon is involved in generating action potentials and in propagating or moving the action potential from the cell body out to the end of the axon. And so this is going to be the part of the neuron that generates a message and then transmits a message. This is the axon hillock, where the cell body actually narrows down to the axon. This is going to be the trigger zone for action potential generation. And so if you'll recall from the last chapter, we talked about action potentials and we talked about a threshold stimulus. And so the threshold stimulus was one where you reach negative 55 millivolts. Now, this point, the trigger zone, is where getting to negative 55 really, really counts. If you hit negative 55 way out on a dendrite on the opposite side of the cell body from the axon hillock, you may or may not get an action potential out of it. But if you hit negative 55 at the axon hillock, it's going to trigger an action potential that will move down the entire axon. So this is sort of the really important part of the axon for generating an action potential. Axoplasm is basically cytoplasm in the axon, and that does play a fairly important role, which we will discuss in more detail probably in the second or third PowerPoint of this series. The axolemma, which I've got these arrows pointing to, is the membrane of the axon. And so with the skeletal muscle chapter, one thing that I hope that you bring forward from that is that events that happen at the surface of the cell membrane or at the cell membrane are really important. This is where your ion channels are, and this is what your ion channels allow ions into and out of the membrane. And it's when you change the charge on that membrane that these cells can carry out their physiological functions. And so the axolemma is the membrane of the axon, and it's an important part of what we're going to see. Now, these are the nodes of Ranvier. You may have noticed that the position of the arrows didn't change, and it didn't change because the nodes of Ranvier are actually areas where the axolemma is not covered by these other types of cells that you're going to see here. These are Schwann cells, and so basically, Schwann cells are responsible for a much faster type of nerve impulse conduction called saltatory conduction. They also contribute to peripheral nerve repair. We'll see that in more detail a little bit later on in this PowerPoint. These Schwann cells are sometimes also called internodes because they are between the nodes of Ranvier. So where the arrows are pointing, each arrow is pointing to one Schwann cell. And each Schwann cell also gets called an internode because each Schwann cell is between two nodes of Ranvier. This portion is the axon terminal, and this is the portion of the cell that is secretory. So this is where neurons actually produce the neurotransmitters, or not produce them, I'm sorry. This is where the neurons actually release the neurotransmitters that are responsible for communication between neurons and other cells. And so let me backtrack for just a minute and say that the cell body is actually the area of the cell where these neurotransmitters are produced. And then after they're produced, then they migrate down the axon to the axon terminals. So now we're going to take a look at some of the structural classes of neurons. There are 3 structural classes, and we're also going to take a look at their 3 functional regions. So the first thing to say is we're going to be making reference to processes that come out from the neurons. So you'll remember from the skeleton that a process was a projection of the bone, of bony tissue up from the bone. And in this case, a process is a projection of cytoplasm and cell membrane out from the cell body. And so your neuron types are going to make a reference to the number of processes that are actually coming out from the cell body. The 3 functional regions are going to be receptive, conducting, and secretory. And so the main thing that I want you to pay attention to here with these regions is that they contain different types of membrane proteins. And so your receptive region is going to consist of membrane proteins that have the ability to receive signals. Now, a lot of these signals are going to be chemically gated channels, chemically gated ion channels. So this is something that you saw in the motor end plate in muscle tissue. A channel that opens up in response to some sort of signal or neurotransmitter binding to it. There are other varieties of ion channels in this region, but we'll discuss those in more detail, I believe, in the second PowerPoint in this series. The second region is going to have your voltage-gated sodium and potassium channels. So these are channels that hopefully you're already a little bit familiar with, the same types of channels that transmit action potentials across a skeletal muscle cell are actually going to be the same type of voltage-gated channels that transmit an action potential down an axon. So that's going to be the conducting region where you find the voltage-gated sodium and potassium channels. So if you'll recall from the last slide when we talked about the axon hillock and called it a trigger zone, the axon hillock is actually the area of the neuron where you transition from chemically gated ion channels to voltage-gated ion channels. And so these different areas of the neuron are going to respond to different types of stimulus in terms of opening their gates up and letting sodium in and potassium out. So the chemically gated channels in the receptor region, you can hit them with as much of a voltage change as you want to. They're never going to open. That's not what they respond to. And likewise, with the conducting region, you could take an axon, which is where the conducting region is, and you could dip it in a bucket of neurotransmitter, and nothing is going to happen because it does not respond to chemical signals. They only respond to voltage changes. And what this means is that the different areas of the cell actually have completely different functions. One is going to be to receive signal, and the other one is going to be to generate and transmit an electrical signal. So I just wanted to say that this is where all of the talk that we did about moving things around in the cell actually makes a difference. So you make a particular type of protein, and then you have control over where you move it to in the cell. And it's the ability to send proteins to a very specific destination that allows for multiple functions like this, for the same cell to be able to part of the cell to receive a chemical signal, for a different part of the cell to generate and send an electrical signal, and for the third part of the cell, the secretory region, to actually release neurotransmitters. And so the reason why you've got three functional regions here is because the different parts of the cell have different membrane proteins which do different things. Okay, so the first type of neuron that we're going to talk about is the multipolar neuron. And the first thing that I'm going to tell you about these terms is that you can think of polar as meaning one pole being one process. So multipolar means multiple processes. And so if you look at the cell, you can see that there's one big axon coming out, and there's never going to be more than one axon. There's going to be one axon coming out, and there's going to be multiple dendrites coming out from the cell body. And so they're with multiple processes, multiple poles, multipolar. If you take a look at the functional regions here, you can see that the dendrites and the cell body are actually what make up the receptive region, and it's the axon hillock and the axon which make up the conducting region. So the blue portion here is where you're receiving the chemical signal, where you have those chemically gated ion channels that can actually respond to the chemical signal. The pink area is where you generate and transmit an action potential. So this is where you have membrane proteins that can generate and transmit electrical signals. And in the green region, the third region here, this is where you actually release neurotransmitters. You're going to have voltage-gated calcium channels because it's necessary for calcium coming into that region. Calcium is an absolute requirement here for secretion of neurotransmitters, and we'll get into that in a little bit more detail later on. This is a bipolar neuron. So here you can tell that there are two major projections coming out from the cell body, just two of them. So two projections, two poles, bipolar, one big dendrite, one big axon. So this is usually where in class I have somebody ask me if bipolar neurons have anything to do with bipolar disorder, and the answer to that is actually no. So the name is just basically a coincidence. Okay. These are the three regions in a bipolar neuron, and I want you to notice that the point of commonality here between multipolar and bipolar is that the bipolar has a receptive region involving dendrites and the cell body, and a conducting region involving the axon. And so one thing that I hope that you notice here is that there's a pretty conspicuous absence of Schwann cells in the bipolar neurons. And there are two reasons for that. First of all, you only find Schwann cells on neurons that are in the peripheral nervous system. But secondly, most bipolar neurons are really short. So this means you don't particularly need to speed up the nerve impulse transmission. And so I'll make more reference to that later on. So this is the difference between being what's called myelinated and non-myelinated. Okay, but the biggest thing that I want you to notice here is that, again, receptive region in the dendrites and cell body, and the conducting region with the axon, secretory region at the axon terminals. This is our third type of neuron, and this is going to be a unipolar neuron. So in a unipolar neuron, you've actually got just one projection coming out from that cell body. So you'll notice there's that rounded cell body, one projection coming out from it. And that projection is going to split off in two directions. One is going to be the peripheral process, which carries signal from the receptive endings toward the central process. So actually in this case, the receptive region is going to be way out here at the end of the peripheral process. And you'll notice that the conducting region actually goes right by the cell body. So in this case, the cell body isn't part of the receptive region. It kind of gets bypassed by the signal going from the peripheral process to the central process. There's a secretory region where you have axon terminals releasing neurotransmitters. And so one thing that I wanted to tell you about the unipolar neurons is that some of them can be really long. So these tend to be sensory neurons. And with most sensory neurons, you have some sort of receptive region where you're sensing whatever stimulus is going on. So let's say you stub your big toe. So you're going to have sensors in the skin right there. Now, the secretory region is actually going to be embedded in the spinal cord. Now, one thing that may surprise you is that the spinal cord actually ends around L1 or L2. And so that's actually above your hips. So this is above the sacrum, and it's the first or second lumbar vertebra. So this is actually superior to the hips. That's a long way for a cell to travel, but the peripheral process actually goes from your big toe, in this case, all the way up to just outside the central nervous system. So your cell body is going to be located in a group of cell bodies that are fairly close to the spinal cord. And so this signal goes a long way. So these conducting regions can go quite a ways. I kind of want you to think about how long one of these neurons would be in a giraffe, where in the foreleg of a giraffe, where I don't know if it feels some sort of pain in its hoof, how long that signal has to travel, but these signals do travel a good distance. Okay, so you can see a lot of variation in these structural classes of neurons. So if we take a look at the multipolar neurons, you can see that you've got a lot of dendrites that are branching out, but the Purkinje cell of the cerebellum on the left and the pyramidal cell on the right, they look very different. But the fact that they've all got multiple processes coming out from the cell body means that they are indeed multipolar. So dendrites and axon. These are bipolar neurons, and on the left, the olfactory cell, even though that dendrite looks awfully thick and makes the whole thing look a little bit like a hydra, the dendrite, there's one dendrite coming out and there's one axon coming out. So you can see here that these are both definitely bipolar cells. And in the unipolar neuron, which is all the way on the right, you've got one projection that's coming out from the cell body. So these neurons can look very different, but you're still going to find just three major structural classes. So what is it that they actually do? The multipolar is by far the most abundant structural type of neuron in the central nervous system. And the main reason why is because these are the neurons that are present in your spinal cord and in your brain. So this, with this being the major neuron type in the central nervous system, that's actually the majority of neurons in the body. Bipolar neurons are rare. They're found mainly in special sensory organs. So the mucosa, olfactory mucosa, so this is going to be neurons that are involved in the sense of smell. They're also going to be found in the retina of the eye. And they're also going to be found in the vestibulocochlear organ in the inner ear. So unipolar neurons are found mainly in the peripheral nervous system, and you usually just find these, they say, they're common in the dorsal root ganglia of the spinal cord. We'll take a look at that toward the end of this particular chapter, what the dorsal root ganglion is, but you're going to find these in structures that approach the spinal cord and in nerves that are coming into the spinal cord and into the brain. So if you look, there's just the one peripheral process still. Okay, now if we classify neurons by function, you're going to have sensory or afferent neurons, and again, these are going to be responsible for bringing information into the inter or association neurons, which are usually involved in either integration of signal, in other words, assembling a lot of different signals into one bigger picture, or association neurons, which transmit signal from one neuron to another neuron as you're moving from the peripheral nervous system to the central nervous system. And so basically, you've got sensory or afferent neurons that pick up stimuli, bring them into the inter or association neurons. These are the neurons that usually identify the stimulus and make some sort of decision about the stimulus, and then they're going to send directions to the motor or efferent neurons. Okay, so again, what are most of our different types of neurons involved in? Most multipolar neurons are interneurons or association neurons. So again, these are going to be responsible for integrating sensory input, for sending directions in terms of motor output. They can be a part of a chain of neurons. So it usually takes 3 neurons to get from a receptor out in the skin up to the appropriate part of the brain. It usually takes, sorry about that, that was a phone call that was coming in. So where was I? Okay, so as I said, it's usually a 3 neuron chain that gets you from some sort of sensor up to the central nervous system, and it's usually a 2 or 3 neuron chain that gets you from the central nervous system out to some sort of effector. And whenever you've got these neurons that are in these multiple chains of neurons that are carrying a signal, these are going to be multipolar neurons. Some multipolar neurons are motor neurons, and in a lot of cases, as you can see from the diagram below that, you're going to have a neuron that's embedded in the spinal cord and actually goes all the way out to that particular effector. Now with this diagram, I'd like you to sort of pause for a second, and you can pause my talking. I know you'll be just horrified to do that. But what you can see here is that you've got a label that points to the interneuron or the association neuron, and you've got a label that points to the motor neuron. And what I want to point out there is there's a dot and a line with the interneuron. And so the dot is going to be the cell body, and the line is generally going to be the axon. If you look at the diagram all the way over to the right with a sensory neuron, you see a line, and then you see a dot at the end, like up towards the top. You see this little branch coming out of the blue line that's got a dot on the end of it. That dot's going to be the cell body, and so the fact that you've got this one thing coming off the cell body means it's going to be a unipolar neuron. So back again to multipolar neurons, so most of them are interneurons or motor neurons. Bipolar neurons are sensory neurons, and we talked about that previously. Most unipolar neurons are sensory neurons, and these can be called first-order sensory neurons, the ones that directly are in contact with the stimulus and take it back up to the spinal cord. Okay, so again, closer look at the multipolar neurons, and this is where you can actually have a closer look at this diagram. And again, just looking at the interneuron or association neuron, look at that green neuron. You see a dot on the end of it. That dot represents the cell body. The line represents the axon. And the little fork at the end of it represents the axon terminals. So when you're looking at these diagrams or these sort of maps of neurons, that's how I want you to interpret these. That the dots are the cell bodies, that the lines are the axons, and that the forked regions are actually the axon terminals. And hopefully that makes a little bit more sense, because you can see that the axon terminals of the green neuron are right up beside the cell body of the red neuron. And so the fork being a transmitting region, the circle being a receiving region. Hopefully that makes sense. Again, this is a closer look at bipolar neurons. You're going to find those in the retina of the eye, in the inner ear, and in the olfactory mucosa. This is the sensory neuron that I was talking about. And so again, if you'll notice up to the top, you'll see that there is a little fork in what I've called the axon. But actually, that much smaller branch is where the process actually comes out from the cell body and splits into the peripheral process, which goes all the way from the skin up to where that first process is, and the central process, which goes from that first process up to where it forks into the spinal cord. Okay, so that's basically our talk on neurons. The next thing that we're going to talk about is neuroglia, which are sort of support cells for the neurons. And these make up about half the volume of the brain. So they're much, much smaller than neurons. And one thing that I want to kind of bring back to your memory is looking at that neuron smear that we did very early on in lab this semester. And so you had the neuron, which was this huge cell, and you had the neuroglia, which were around it, which were much, much smaller. Okay, so the neuroglia are much smaller than the neurons, but since they make up half the volume of the brain, they're actually way more numerous than neurons. So unlike mature neurons, neuroglia are going to carry out cell division, and this is the reason why they're the only type of cell that's actually responsible for brain tumors. So the thing is, with the neuroglia, you don't have any important role in the generation or the propagation of action potentials. And so there have been occasional exceptions discovered, but for the most part, the neuroglia aren't really involved in communication. They're involved in support roles. And so your types of neuroglia actually differ from the central nervous system to the peripheral nervous system. So we talked about the difference between central nervous system and peripheral nervous system actually being brain and spinal cord versus everything else, but it is more than just a locational division. They've actually got different types of cells, and therefore are almost like two different types of tissue. So the central nervous system has 4 different types of neuroglia. They're going to be ependymal cells, and ependymal cells, we'll see more of their role when we talk about the central nervous system. The ependymal cells are involved in basically filtering cerebrospinal fluid from blood plasma. Oligodendrocytes are responsible for myelination of neurons in the central nervous system. And so we'll talk about that more in just a minute. Astrocytes are star-shaped, and that's the reason for the name. They've got a lot of different functions, which we'll discuss in a minute. And microglia, which are going to be a little bit smaller, and they're not going to look quite as star-shaped. So again, we'll discuss the functions of those in just a minute. The peripheral nervous system has got 2 different types of cells in it. Satellite cells, which you can see are on top of a unipolar neuron here. So their job is basically kind of 2 parts. They can supply nutrients to surrounding neurons, and they can also kind of hold the neurons in place. The second type of cell that we'll talk about is Schwann cells, and these have a really important role in terms of nerve impulse transmission and in nerve repair in the peripheral nervous system. So let's go through the neuroglia of the central nervous system. And so I want to point these out to you. As you look at the left column, you're going to see the oligodendrocyte, and that's sort of a pinkish color. And you can see that stretching out projections that actually surround the axons of the neurons. And so their job is going to be to cover the axons of the neurons with a substance called myelin, and we'll discuss the importance of that a little bit later on. You can see the astrocytes in blue, and you're going to see here that those astrocytes are going to be reaching out processes to both capillaries and to neurons, and I'll explain the importance of that in just a minute. And the microglial cells, that's going to be the 3rd term down on the left, that's in green, and the microglia are usually involved more in phagocytosis than anything else. So, and at the very bottom here, you see the ependymal cells with cilia on them, And again, they're responsible for producing cerebrospinal fluid. So the functions of astrocytes in more details. So for one thing, they provide structural support for neurons. So I want to remind you that in synapses, that axon terminals don't physically contact the cell that they're sending neurotransmitters to, but they're in very close proximity. And this means that the actual physical location of the neurons can be pretty important. And so these astrocytes can provide structural support for neurons, keeping them in the right place for them to be able to efficiently communicate with one another. They've also got a role in the blood-brain barrier. And so the processes that are associated with the capillaries, and you can see that this cell on the top right is sending out processes to both capillaries and axons. And so the processes that they're sending out to the capillaries, you'll see that they're very tightly joined together, and this decreases the permeability of these capillaries. And so this makes the blood-brain barrier a good deal more efficient. So before we go on to the role in learning and memory, I want to talk about this blood-brain barrier in a little bit more detail. So one thing that we have discussed about capillaries, at least in passing in lecture, is that they're leaky, that the pressure from the heart can actually force fluid out of capillaries into the surrounding areas around the capillaries. And what happens here with these astrocytes is that they are covering the capillaries so efficiently that they have a lot of control over what can actually leak out from those capillaries into the surrounding tissue, which is the central nervous system. And it basically gives your central nervous system a physical barrier that keeps a lot of materials from getting across them. So the astrocytes really have a greater deal of control over what goes into and out of the central nervous system. Now, most of the time, this is a really good thing, because if you're exposed to some sort of toxin in the environment, if it somehow makes it into your bloodstream, there's much less of a chance that it gets into your central nervous system. But one of the biggest problems that you run into in terms of medicine is when you have something that's going wrong with the central nervous system, and it's pretty difficult to deliver a medication to the central nervous system. And the classical example of this is Parkinson's disease, where cells of the central nervous system don't produce enough of a neurotransmitter called dopamine. And so the first time that I heard that, I thought, well, why don't they just put some dopamine in an IV and put it and stick it into somebody's circulatory system? And the reason why is because the blood-brain barrier blocks the passage of dopamine from the circulatory system into the central nervous system, so you can't address it like that. There is a synthetic form of dopamine that can cross the blood-brain barrier, but unfortunately for Parkinson's patients, the blood-brain barrier tends to be adaptive, and so things that can make it through early on in the treatment of the disease don't make it through later on. So the blood-brain barrier is able to filter out a lot of chemicals, to keep a lot of chemicals from making it into the brain, and it is not only pretty good at what it does, but it's also adaptive at what it does. And like I said, under most circumstances, this is a really good thing, but when you're talking about therapeutic measures, it can be a real hindrance. Okay, astrocytes also have a role in learning and memory. That role isn't clearly defined, but when you have abnormal numbers of astrocytes, you do see problems with learning and memory. Astrocytes also maintain the chemical environment, and so this right here should tell you the ways in which they exert control over the blood-brain barrier. They regulate the ion concentration in their surrounding environment. They take up excess neurotransmitters that provide nutrients to neurons. They're also joined by gap neurons, so gap junctions, excuse me, so they can pass materials from one to another pretty easily. So this is the next thing that I want to talk about, where it says take up excess neurotransmitters. So in a lot of cases, clinical depression is caused by a serotonin shortage. And in many cases, the serotonin shortage can be where the astrocytes are too active in taking up serotonin. So they're taking up so much serotonin that the levels of serotonin drop below what they should be, and this can result in pretty severe depression. And so Things like sertraline, Zoloft, Prozac, a lot of these are serotonin reuptake inhibitors, and what they're going to do is to either block or cut down on the action of these astrocytes in picking up serotonin. So if the reuptake of serotonin is inhibited, it means your levels of serotonin in the central nervous system rise to what would be more normal levels. Now you have to be very careful to not overdo it with the suppression of these astrocytes, because a deficit of serotonin leads to clinical depression. A surplus of serotonin can lead to depression and schizophrenia, and a big overload of serotonin can cause what's called serotonin syndrome, which can kill you as quickly as 48 hours. They also have a role in embryogenesis of the brain, and like the role in learning and memory, this is not well defined, but when there are abnormal numbers of astrocytes, then the brain does not develop normally. Okay, microglia they're small cells. That's the reason why they get called micro. They've got a lot of projections to them, and what their job is basically phagocytosis. And so they're going to remove any sort of cellular debris, and there's going to be a lot of that generated by neurons because their metabolism runs so high. They have a role in embryogenesis, and as with astrocytes, it's not that well known, but again, if there are abnormal numbers of microglia, then embryogenesis doesn't happen normally. They're responsible for cleaning up after nervous tissue damage. So if somebody has some sort of damage to neurons, then there's going to be cell fragments lying around, and they're going to clean up after that damage. And they're also going to go after microbes. Now, one thing that I want to emphasize is that microglia are not very good at this job. Your normal bacterium can just pretty much laugh off a microglia, and there's sort of an analogy that I want you to remember. So your white blood cells are usually what combat microbes, and they're very good at their job. And I want you to think of them as basically like if you're thinking of a medieval siege of a castle. If your body is under siege by microbes, then your white blood cells are the really highly trained warriors that go out and that fight off the people that are sieging the castle. The microglia are kind of like when you've sent out all the warriors and you've got your last ditch defenders of the castle. You know, give a 12-year-old a spear or give a 70-year-old man a spear and say, "Here, defend the castle." That's kind of like what microglia are. So even though they can do some damage to microbes, they're not that great at their job. So this is one of the reasons why an infection of the central nervous system can result in sky-high fevers. It's because the central nervous system knows that the microglia are not really up to this task, and that's the reason why bacterial meningitis can result in incredibly high fevers, because the fever is a better defense for the central nervous system actually than the microglia are. Okay, so these are oligodendrocytes, and as you saw in a previous graphic and also in this graphic, what they're doing is they myelinate axons in the central nervous system. And so basically, they send out cytoplasmic processes that will wrap around the axons of neurons. And basically, what this myelin does is to insulate axons. The main importance of the myelin is that it speeds up nerve impulses by about a factor of 50. So wherever you want much quicker communication with neurons, they're going to be myelinated. And so when I put out a lab PowerPoint, you'll see in that lab PowerPoint that there are white and gray. There's white matter in the brain, there's gray matter in the brain. And so the white matter and the gray matter in the brain are actually places that have myelin or don't have myelin. Okay, so oligodendrocytes can myelinate more than one axon at the same time, so they can reach out processes that myelinate multiple axons. Okay, they don't play a role in any kind of damage repair, and that may have come out of left field, but when we talk about Schwann cells, I think that you'll understand this a bit better. So Schwann cells are basically kind of like the oligodendrocytes of the peripheral nervous system, except what they don't do is to reach out processes to multiple axons. What they do is you'll have one Schwann cell that rolls around an axon. And so basically, Their job is to synthesize myelin, which speeds up nerve impulse transmission. So the way that it does this is to coil around it in kind of a jelly roll fashion. And what I usually remind students of when I'm lecturing is like Hostess Swiss rolls, those chocolate rolls, and if you bite into it, you'll see that sort of spiral of white icing on the inside of it. That's kind of the picture that I want you to have here with the Schwann cell. So they circle around this myelinated axon. Now, if the axons are in a clump, um, then they'll surround it. They won't do too much coiling because they have a lot more space to go around. But the axons have to be bundled in order for them to do this. They can't reach out processes to multiple separate axons. Now, Schwann cells are involved in the repair of damaged peripheral nerves. So, if you have some sort of injury to the spinal cord that's very difficult to heal, but you can get healing in damage to nerves in the peripheral nervous system. And here's the reason why. So when damage happens, what's called axonal dieback will occur. And if you look at the neuron here, you can see that the axon actually dies all the way back up to where the cell body is. Now, if you look at the Schwann cells here, they've maintained their position, and they actually maintain a hollow space within themselves called an endotube. And what they'll do is they'll produce a signal that can act as a guide for new axonal formation. And so if you look at the diagram again, you'll see the axon growing its way back down. And this repair actually occurs at about the length of 1 millimeter per day. This is the reason why peripheral nerve repair or regaining function of peripheral nerves can be so dang slow, because this regrowth happens really slowly. But the thing that is most certain is that Schwann cell myelination is an absolute requirement for nerve regeneration. Where you've got Schwann cells, you have a possibility for nerve regeneration. Where you have oligodendrocytes, you don't. Okay, so in our next video, we'll take a look at nervous system physiology.